Thanks, everyone. And today I'm going to talk about my research about imaging research. So the title is Development of PET Neuroimaging Probes Targeting Epigenetics. My name is Changning Wang. I'm from Martinez Center for Biomedical Imaging at Mass General Hospital. So today I want to quickly go through the PET, the positron emission tomography first, then show some examples of my research target epigenetics, for example, the HDAX and the SERTIN1. So why we focus on the PET imaging? So here is a review and showing the imaging modalities we have now for both clinical and preclinical use. On the top part of this figure, so there are kind of X-ray or CT, PET, MRI, ultrasound, etc. And then most timely for both clinical and preclinical research. And the bottom part, basically the optical imaging techniques, which can be used for like body surface imaging or some like organ ex vivo tissue study. Among all these techniques, PET imaging has some advantages. For example, they have really easy to label such small molecules or peptide to study the drug PK or PD. And also, if we have the imaging probe for a certain target, we can see this target density or activity in vivo, for example, the HDAX. Also, because we inject tiny amounts of the PET tracer, normally in nanograms, so we can easily translate the PET tracer to human, compare with the regular therapeutics. That will be easy. For PET imaging, we have to inject some like radioactive materials. We call it imaging probes or radio tracers for in vivo protein or target or receptor or other target expression function in the body. So it starts from like isotope production from a cyclotron, then we go through some chemistry pipelines, get the tracer, then inject to human or animals for imaging and the data analysis. So the two main application of PET tracer is to measure the density difference. For example, you have a patient compared with control. So you want to see the regional difference of this target, we can use PET. Also, we can measure the target engagement with drugs if we have this PET imaging probe for the same target of this drug. So here is like a basic procedures. For example, if we use cyclotron to generate like a half-life or the short half-life isotopes, like a C11, 20 minutes, or F18, like 109 minutes, or nitrogen 13 or oxygen 15, even shorter than 10 minutes. Now, from the cyclotron, we have the high beam proton here and react with the nitrogen in the air to release the helium particle plus the C11. C11 is unstable. It further reacts with the oxygen in the air, so form the CO2, carbon 11 CO2. Then we go through like a chemical conversion from CO2 to different chemical modalities. For example, from here, we can get the C11 methyl iodide, which is most commonly used radioisotopes for chemical reaction. And we labeled with one compound, introduce this C11 to the molecules, then we get this final product in a very short time, normally 3 to 5 minutes for C11. And we need to go through the HPLC purification and some formulation and QC. For the human imaging, we need to get a GMP lab, do the tracer and release those before inject. And then we can inject to the human and put in the scanner to collect the gamma rays from this positron. Then we can get more images. The whole process maybe take like 3 to 4 hours, like 20 minutes or 40 minutes for radioisotope production, then go chemical steps, maybe 40 to 60 minutes. Then we can scan like 1 to 2 hours. So we have like a major PET tracers in two catalogs. For example, we have the receptor or ligand-based tracer. For example, the Radioactive is for dopamine receptor imaging. And also the FDG is for like a glucose metabolism study that's kind of like enzyme or substrate based to measure the activity. There are many drugs. For example, this table lists some like FDA approved drugs for different classes. The number is high here, but compared with the radio tracer, still a lot of space for us to find new tracers. So there is some table summarized currently used in human, some brain radio tracers. That's like roughly like 37, maybe more now, and with 100 molecules each can be used for the human. So compared with the drug target, it's pretty small number. So we try to get more imaging probes for study. And so far, FDA has approved some PET tracers. So they are on market, for example, the FDG or 3 A-beta tracers and 1 tau tracers. And the L-DOPA approved 2 years ago, and some other C11 or F15 tracers or gallium-68 tracers, and some other nitrogen 13, etc. tracers. Totally 13 tracers has been approved by US FDA. Now they are on market for study. So back to my research, epigenetic is one of my main research direction, and I will show you some like basics and the application of the probes. For example, epigenetics, I think people know this like functional modification to genes, but they didn't change the DNA sequence. So we need some technique to see such epigenetic changes in vivo. So we think PET is ideal tool like to do this. And also we can use imaging tool not only for research, also can help the drug development. So we basically need a team with like expertise in probe design, like chemistry, like medicinal chemistry or radiochemistry, and also need like a preclinical study and the translational imaging study. Now the final goal is to go human. There are 2 main components of the epigenetic. One is DNA methylation. The other is like histone modification. Now, for this one, like we have like a reader, writer, eraser for this epigenetic process. Now, HDAC here is kind of eraser to remove the acetyl groups from the histones. Now, like basically 5 classes of HDACs. Class 1 to class 4. Now, class 3 is the special one. They have like 13 from 13.1 to 13.7. The other isoforms we name from class 1 until class 11. So there are functions, for example, in the brain. So there are many studies reported. Now, the HDACs has an important function for like Alzheimer, Huntington, Parkinson, MS, or ALS. So we need to see such changes in such patient populations now if we have the PET imaging tool. Similar to a drug discovery pipeline. So for the PET imaging probes, we start from a lead compound. We need to design a compound with high binding affinity. Then we go through like radiochemistry to think a better and simple way to introduce the radioisotope to the small molecules, peptide, or proteins. We evaluate in animals. Most of the time, we start with rodents. If we do the brain imaging, we need to focus on the brain region. Then we go to the non-human primates before we can go to human. So like we need some like imaging facility. For example, the radiochemistry, that's an automation system. We don't need to control by hand. We just use computer to get some program done, and we use this kind of machine to do the whole radio labeling automatically, to avoid any radiation exposure. Then we can use like a small, we call it mini PET CT for the rodent scan, and also at our center we have like PET MR scanners for brain imaging and whole body imaging. And also we have some new methodologies to radio label the carbon 11 or F18. These some examples we can use more method and a new method to introduce the isotopes. So the example here is called Martinostat. This is the first epigenetic PET imaging probes. Actually, it's a class 1 HDAC probe. And we started this project kind of like from 2007 until 2014, we get the IND done. So we evaluate thousands of compounds and radio label some of them. And because we focus on the brain, so we need a very good high brain uptake tracer and with good PK and BLD, no tox and good specific binding. So, okay, what's next? Okay. So once we get IND, we can move forward to the human step. So
I request you all to hold on for a minute. I will contact Dr. Wang and get back to you.
yeah, sorry guys, I don't know why it's disconnected. Let me continue on my slides. Okay. So here we try to label some like known HDAC inhibitors, for example, this MS275. and we didn't see the brain uptake. So we use like rodents and monkeys to see. So the black region here is PET, the brain region, but it's no signal. That means no uptake here. And so we modified structure and they introduced the C11 to different similar structures. From here, you can see some are with better brain uptake, but some are still low brain uptake. After a while, we many years study, we try, finally we get this tracer, we call it martinostat with some like components, can labeled with C11 easily and with hydroxy group for the zinc binding, also has an elemental group. So to balance the whole log P to let the molecule get in the brain. This is a monkey study. So we can see the brain uptake and also the major organ uptake is good. So what's the requirement for the radio tracer? So first of all, it's need a high binding affinity. Also, we have a tracer, we have the some measurement for Bmax KD at least 5 to reach enough binding potential of the target, and also no off-target binding. That means we evaluate the compound with the commonly studied CNS targets, and no significant inhibition at 10 micromolar concentration. And the simple chemical reaction, so we can react 3 to 5 minutes, forget this tracer done. So all the chemistry are simple here. And the physical binding, normally we test in the disease models, or we can treat the drugs. We call the blocking study compared with baseline, means we only inject the tracer. So we can see the dose-dependent decrease of the uptake. That means the physical binding is good for the tracer. We also test not only in brain, also test in the organs. You can see the blocking, the same blocking. Finally, this is the first human study with this tracer. This is the PET MRI. We can see the distribution of the PET signal. That means class 1 H-tag expression is high in the gray matter, but not in the white matter. That also matched the in vitro tissue study. Then we can do some kinetic modeling analysis for radio tracer before we can validate use in the patient. Some examples of this tracer, for example, we can compare the age difference and the gender difference. So the red color highlighted the increased high uptake of the tracer. That means more H-tag expressed here in the male, and compare in the age. With the age increase, some regions of H-tag expression are increased. The gender difference is like red color showing the females has more than males, and the blue color highlighted the H-tag has less in females compared with males, such as the cerebellum. For the patient study, we can see this is the bipolar patient. Now the blue color means the H-tag expression is low in these highlighted regions, in bipolar patient. And for the drug evaluation, for example, we have another H-tag inhibitor, we call the CNF7, that can get in the brain. We can compare the target engagement in different brain regions. The baseline is tracer only, and if we can inject the drug at 0.5 mg per kg, like 5 minutes or 1 hour before the tracer injection, now we can see the signal decreased. Then we can calculate the target engagement of this tracer. Another example of H-tag family is the SIRT1. So basically, we start with the known structures of the SIRT1 modulators, either binders or inhibitors or activators. Now we found like EX527 may be a good one to start with. Then we can radio label this compound, evaluate in the rodents first. You can see that in the we call the baseline study, the uptake in the brain is pretty good. Then we can treat some drugs. For example, the 5 mg per kg treatment of the unlabeled tracer, and we can see the decrease of signal. That means the specific activity is good. So we also do some like monkey scans in baboon, so you can see the uptake in different regions. Some region high, some region low. You can compare with the time activity curves here. Also, we can track the metabolite corrected plasma. We see this fast washout from plasma. That's good for the tracer, but maybe not ideal for drugs. And parent fraction, that means the parent tracer left in the brain, in the plasma. And so it's pretty good, still has like more than 20% parent compounds at the end of the scan and 90 minutes. So because the ST1 has an important function in the Alzheimer's, for example, we test some AD models. For example, this wild type in the blue shape, and the right shape is the 5XFAD AD mice. So we can see the decrease of the ST1 expression by imaging that match the reported results in vitro by other groups. So for next step, for this tracer, the same for other radio tracers, we need automation in a GMP lab and do more tissue, disease model study and test more in the monkeys. Then we can apply the IND for human study. Thanks my lab members and collaborators and the patient and the families. And thanks for the research support for my project. Thank you for everyone. Glad to take any questions. I will stop here.